Hello everyone, this is Yuxuan Meng. The title of my project is The Incidence of Azole Resistance among non-albicans Candida species, and my supervisor is Dr. Parker. So let's get started. Introduction. Candida albicans and related species are fungi that cause infections, and the resistance to the drugs that treat them is growing. An introduction divided into three parts, which includes the current situation of Candida species infection, how azole work as an antifungal drugs, and the problem of antifungal resistance. So my project is going to focus on the ERG11 mediated resistance to the azole, and the rest of my presentation will explain how to investigate whether this type of resistance is increasing in other Candida species. And Candida species infections, and globally, Candida species infection is one of the most common causes of fungal infections in humans, and leading to more than 1 million deaths annually. About half of the Candida infections are caused by the Candida albicans, whilst the infections caused by the non-albicans species are emerging. For instance, C. auris. And the prevalence of different Candida species infections varies geographically, as can be seen in the figure on the left-hand side. And the spectrum of the disease of the invasive candidiasis ranges from the minimally symptomatic disease to the systematic disease, including species sepsis, with associated mortality exceeding 70%. And notably, nearly 90% of C. auris isolates are resistant to the fluconazole, and which is a frontline antifungal drug. The antifungal drug. The class of antifungal drugs, azoles, exert fungistatic activity by inhibiting the cytochrome P450 enzyme, lanosterol 14-alpha demethylase, encoded by the ERG11 gene, which blocks the ergosterol synthesis, and the azole reduce the amount of the ergosterol and result in the formation of a toxic sterol, as seen in the figure, both of which disrupt the membrane and cause the fungistatic effect. There are two types of the azoles, imidazole and triazole. First generation of the triazoles include the fluconazole and itraconazole, and second generation triazoles such as voriconazole and posaconazole are more effective in treating fluconazole resistant pathogens because they have a higher affinity for the enzyme than fluconazole. Here, the circle on the skeletal formula of the fluconazole and the posaconazole, which I pointed out specific nitrogen atom that binds to the heme of ERG11 enzymes, and this means the rest of the drug contacts different parts of the enzymes, which is why different mutations cause the resistance to the different drugs. Antifungal resistance in the Candida species. The drug resistance results in the normal dose of drug being ineffective in treating the infection. An antifungal drug resistance can arise from the prophylactic drug use. It is indicated by the elevation of minimum inhibitory concentration of a drug, and the most common mechanism of azole resistance includes amino acid substitution in ERG11, which decrease the drug binding affinity. And different azoles will result in different minimum inhibitory concentrations in different candida species, as they will have different affinities for the specific drug target, and this can be seen in the table for fluconazole and posaconazole. For 3 species of the candida, resistance leads to the failure of the treatment and can occur through the several mechanisms, but for my project, I will be focusing on ERG11 point mutations. The ERG11 point mutation, and there are over 140 amino acid substitutions in ERG11 which have been found to be associated with the azole resistance in Candida species. And most of these substitutions are clustered into 3 hotspot regions, and some of the same resistance mutations known in C. albicans have now also been identified in C. auris, and especially changes at the tyrosine 132 and lysine 143. And from this image, you can see that I have highlighted the right circle for the tyrosine 132, and yeah, that's the site of the amino acid substitution. Hypothesis. So my hypothesis is therefore the azole resistance amongst the non-albicans Candida species infections is increasing. Objectives. So in order to test my hypothesis, my objectives of my project are to identify the resistance caused by the ERG11 mutations in different Candida species in the literature. And I will correlate the amino acid substitutions in ERG11 and associated resistance profiles, which will cover the differences in MIC for fluconazole, itraconazole, posaconazole, and voriconazole, and compare the non-albicans and the albican species. And I will also relate these to geographical occurrence of the infection, the type of infection, any patient's pre-existing medical condition, the previous treatment, and the date of the resistance. And I will also investigate whether the same mutations are seen in non-albican species as in albicans by analyzing and comparing the wild type and mutated ERG11 sequence from the genome database and the literature. And finally, I will identify the ERG11 mutations in sequence deposited in the genome database, which could also confer azole resistance, but which have not been reported in the literature. And though through this work, I will be able to access whether resistance is increasing in non-albican species. For the project plan, here in details, the systematic literature review will focus on the studies and clinical reports on the Candida albicans and non-albican Candida species, resistance to the identified ERG11 substitution, and the resistant profile of strains. And the literature relevant to ERG11 mutation and amino acid substitution will be used then in conjugation with bioinformatics approaches, and bioinformatic analysis of the sequence of ERG11 enzyme can assist in categorizing and distinguishing between resistance caused by the different point mutations, and newly deposited ERG11 sequences will also be analyzed for the presence of the substitution. and a research strategy will then be developed to express, purify, and characterize the non-albicans ERG11 proteins. And these are my references. Thanks for listening. Have a good day. Bye bye.